So in the previous video, we had stopped at the enzyme Rubisco, which not only helps attaching RuBP with carbon dioxide, but also has an affinity towards oxygen, which results in RuBP reacting with oxygen. So when RuBP reacts with carbon dioxide, it is called the Calvin cycle. And when RuBP reacts with oxygen with the help of the Rubisco enzyme, it is called photorespiration. So let us see what photorespiration is and how it is different from the Calvin cycle. Photorespiration is also a cycle like Calvin cycle, but it is different in the molecules that is generated. So if you see here, this is the normal Calvin cycle and this is the photorespiration cycle. So photorespiration is also beginning in the chloroplast when RuBP will attach with oxygen with the help of Rubisco. While in the place of in Calvin cycle, you see RuBP is attaching itself to carbon dioxide with the help of Rubisco. Here in photorespiration, RuBP reacts with oxygen with the help of Rubisco. Now, in the case of Calvin cycle, we see that 3 PGA molecules are produced. Like if 6 RuBP combines with 6 carbon dioxide, 12 molecules of 3 PGA is formed. But in the case of photorespiration, you see when 6 RuBP combines with 6 O2, we get 2 molecules in place of 1, which is 6 molecules of 3 PGA and 6 molecules of another molecule called phosphoglycolate. Now, this is the first place where the difference is occurring. So, 3 PGA and 6 phosphoglycolate are formed. Now, phosphoglycolate, what it does, now this is an unstable compound and Calvin cycle is only possible, or the cycle will only continue if PGA is available in the cycle. Phosphoglycolate cannot participate in this cycle. So, apart from the 6,3 PGA formed, phosphoglycolate formed will undergo another cycle called the salvage pathway and get converted to 3 molecules of 3 PGA. So, phosphoglycolate during the process of getting converted to PGA loses 3 carbon dioxide molecules. So, what is happening over here is carbon dioxide molecules, which should be ideally available for photosynthetic process, is consumed or is used up in the salvage cycle and is not allowed to participate in this cycle, as a result of which the carbon dioxide molecule is lost and it is wasted. Um, Ideally, in which case it would have been used for the complete to complete the photosynthetic cycle. So during the salvage pathway, which is the transformation of phosphoglycolate to 3 PGA, carbon dioxide molecules are lost and 3 PGA molecules are formed. So now the phosphoglycolate is converted to PGA. Now PGA can take part in this cycle. But unfortunately, if you see the number, instead of 12 3-PGA, phosphoglycolate is only able to produce 3 3-PGA. So in total, we have 9 PGA molecules. Now 9 PGA molecules get converted to 9 G3P. Now if you see here, we see that if 12 G3P is produced, then you can then 10 of it goes in the regeneration of RuBP and 2 of it is helping in the formation of 1 glucose molecule. But unfortunately, since there is only 9 G3P molecules, all of these 9 will go in the regeneration of RuBP, but again 10 molecules of G3P is required for the regeneration of the entire RuBP that was participating in the cycle. But since there is only 9 molecules, all of it will go for RuBP regeneration, but not enough RuBP will be produced because only 5 RuBP will be produced with the help of 9 G3P, not 6 RuBP. So, in net total in this cycle, if you see photorespiration, we see that carbon dioxide is wasted. On top of that, G3P formed is not helping in the production of any glucose, and not only that, it is also not helping in the helping in enough RuBP regeneration to continue this cycle. So this process is called photorespiration, where the RUBP will attach itself to oxygen and undergo this cycle. So if you see, this diagram is a representation of how the phosphoglycolate is getting converted into PGA. So in the Calvin cycle, the RUBP and oxygen, in the RUBP and oxygen will react in the presence of rubisco, form PGA and phosphoglycolate. Phosphoglycolate will get converted to glycolate in the chloroplast itself. Then from there, glycolate will go into the peroxisome, where it is again undergoing some reaction to form glycine. Now glycine will enter mitochondria, and in mitochondria is where the carbon dioxide is released, and this glycine will form something called serine. Serine will come back to the peroxisome. It will get converted to glycerate. Glycerate will again come back to chloroplast, and it gets converted to PGA. So this is the for the cycle that is happening, the salvage pathway that is happening. This is a representation of the salvage pathway, and this whole cycle is a representation of photorespiration. So I put it here so that you can understand what is the difference between Calvin cycle and photorespiration. Now again, initially I had showed an equation to equalize the number of carbons that is formed during the Calvin cycle. So in that reaction, we had seen that RUBP is a 5-carbon molecule. So 6 molecules of RUBP and 6 molecules of carbon dioxide will react to give uh, 36 carbon molecule, which is nothing but 12 molecules of PGA. Now, this 12 molecules of PGA will get converted to 12 G3A, which is consisting of 36 carbon molecule. Now, this 12 G3P will get converted, will be divided into 10 and 2. 10 will go for the regeneration of RUBP, and 2 will go for the production of carbohydrates or glucose. Whereas in the same photorespiration reaction, we see that 6 RUBP reacts with 6 oxygen. But unfortunately, since oxygen doesn't have carbon molecule, there is only a total of 30 carbon molecules that is generated from this reaction. That 30, 6 of them will be a PGA and 6 glycolate will be formed. But 6 this will be 18, and phosphoglycolate is a 2 carbon molecule, so 6 3s are 18 and 6 2s are 12. Totally this combination is giving us a 30 carbon molecule. Now this 12 carbon molecule will again get converted to 3 PGA, and during which it is losing 3 carbon dioxide. So 4 carbon molecules are lost here, 3 3s are 9 carbon molecules are present over here, and this 9 carbon molecule in the form of 3 PGA will be entering the cycle again. But unfortunately here again you see that we only have 27 carbon molecules, 6 into 3 27 and 3 into 3 9, totally 27 carbon molecules. If you see here we have 36 carbon molecules. So for the cycle to be complete, we need 36 carbon molecules, but unfortunately we have only 27 carbon molecules, and in that 27 carbon molecules, none of it will be used for the production of glucose, because there isn't enough carbon molecules for glucose production. And similarly, out of the 27 G3P formed, 25 carbon will be used in the production of 5 RUBP. But again, this 5 RUBP is not going to help in the cycle to continue. So what it will do is, it will take one more RUBP molecule and then continue this photorespiration process, which is again wastage of RUBP. RUBP is not getting regenerated. So this is why we say that photorespiration is a wasteful process in terms of photosynthesis. Now, such a complicated process will not be existing in the plant system if it wasn't useful for the plants. It might be useful for the plants in various other ways, but as if you consider photosynthesis as a process, the photorespiration is wasteful. Now, why? Because there is a lot of carbon loss. Unlike the Calvin cycle, photorespiration is releasing carbon dioxide, and then which could have been used for carbon fixation. Then energy consumption is also there. So if you see here, during all these conversion processes, energy is used, but this energy, if could be used for photosynthesis, the Calvin cycle would have been more useful. And this energy is used, and there is no useful byproducts that are formed. We don't have glucose being formed, RUBP is not regenerated. So, this is in total reducing the efficiency of the carbohydrate production by the plant. So this is just a brief comparison of how the photorespiration and Calvin cycle are varying in terms of what the reactant is. RUBP is reacting with carbon dioxide in Calvin cycle, oxygen in photorespiration. In case of the enzyme, it is Rubisco itself, but the Rubisco is having more affinity for carbon dioxide, and as a result is helping carbon dioxide react with RUBP, but in this case it helps oxygen to react with RUBP. Here sugars are formed, but here 3-PGA, carbon dioxide and ammonia is formed. It's a very highly efficient process, very energy intensive and wasteful process. And the environmental trigger for this is this happens when there is high carbon dioxide and low oxygen. But this process happens when in the environment you find that the amount of oxygen is more than the carbon dioxide. So high oxygen to carbon dioxide ratio is what is triggering this photorespiration reaction. Now this photorespiration, in order to avoid this photorespiration in plants, that is plants evolved naturally so that this wasteful process is eliminated from the system, and this kind of evolution led to the development or the evolution of another type of another set of plants that undergo a different type of photosynthetic cycle called C4 and CAM cycles. So C4 is one category of plants categorized based on their photosynthetic capability. CAM is also a group of plants that are classified based on the different photosynthetic pathway that they follow. So let's see how is this C3 and C4 photosynthesis different, and how are the plant's leaf structure also different. So initially we had looked at the C3 plant's leaf structure, where we see an upper epidermis, we see palisade cells, then there are some spongy mesophyll cells, and we see at the below there is a lower epidermis with a stoma, and the vascular bundles in this form available within the the mesophyll cells. But when we move to a C4 plant, we see that there is a quite different structure. You see the upper epidermis here, and we see that the palisades, there are some palisade mesophyll cells here, and then as we go to this one structure, we see that this is the mesophyll cells. These are the mesophyll cells as well, but here mesophyll cells are arranged in a concentric circular manner over the bundle sheath cells. So if you see here around the vascular bundle, you see there is a bundle sheath cell, but it is of not much prominence over here. But in this case, we see that around the vascular tissues, which is the element flowing, we see that there is a bundle sheath cell, and around that bundle sheath cells you find the mesophyll cell. So this is how the leaf structure of a C3 and C4 plants vary. Now how is this affecting the photosynthesis is what we'll see now. This is also another more different picture of the cross section. We see that this is the vascular bundle, and we see that this is the bundle sheath, and you can clearly see how this is not in green color, indicating that there is not much photosynthesis happening over here in this bundle sheath cell. Most of it is happening in the palisade and the mesophyll cells. But when you come here, we see the vascular bundle, we see the bundle sheath, which is having more pigments, that is, it is green in color, and then we also see the mesophyll cells, which are stacked around the bundle sheath cell, unlike here where the palisade and the spongy mesophyll is in this form. So how does a C4 photosynthesis vary when compared to C3 is what we'll see now. So in C3 we know that in the palisade mesophyll, carbon dioxide is reacting with RuBP, RuBP is converted to PGA, PGA is converted to G3P, which helps in the formation of glucose, and G3P, some molecules of G3P is used in the regeneration. But in the case of C4 photosynthesis, we see that inside the palisade mesophyll, this is not the cycle that is going on. This is not the Calvin cycle that is going on, but instead there is another enzyme here in place of RuBP called PEP enzyme. This enzyme is a PEP molecule. This molecule which is used in place of RuBP is a 3-carbon molecule. So it reacts with carbon dioxide over here because RuBP has a high affinity for carbon dioxide as well as oxygen. But PEP here has only an affinity for carbon dioxide. It doesn't react with oxygen. So what it does is it reacts with carbon dioxide, forms oxaloacetate. Oxaloacetate gets converted to malate, and malate then diffuses into the bundle sheath cell. And here if you see, the malate again reacts with carbon dioxide to form RuBP. RuBP then again takes part in the Calvin cycle, and then when the malate is getting attached with carbon dioxide, one molecule is going for carbon dioxide, one molecule is going in the reaction of carbon dioxide, and the other 3 molecules are used for forming pyruvate. Pyruvate is the one that enters the palisade mesophyll cells, and as a result of which RuBP over here is only able to react with carbon dioxide. So there is no oxygen entry in bundle sheath cell. Even if there is some oxygen in the mesophyll cell, which there will be definitely, PEP is not having any affinity towards oxygen. So it only reacts with carbon dioxide. But while reacting with carbon dioxide, it gets converted to oxaloacetate. Oxaloacetate gets converted to malate. Malate then diffuses into the bundle sheath. One carbon malates one carbon reacts with one carbon is used for the formation of carbon dioxide, and then that reacts with RuBP. Malate is getting converted into carbon dioxide, and that is actually getting that is what is actually reactive with RuBP. So malate is the one that is providing carbon dioxide for this Calvin cycle to happen in the bundle sheath cell. So although the Calvin cycle is happening, it is happening in the bundle sheath cell, and in the cycle for C4 plant, carbon dioxide is not available in the free form to RuBP. It is sent to this bundle sheath cell in the form of malate, where malate reduces to carbon dioxide, and then the remaining carbon molecules forms pyruvate and then diffuses back, sent back to the palisade mesophyll cell. So there is no role for oxygen at all over here, because PEP only attaches to carbon dioxide. So this is another more clear cycle that is shown here. PEP, and the enzyme that helps in PEP to react with carbon dioxide is PEP carboxylase. You see, Rubisco is RuBP oxygenase carboxylate, whereas here this is only PEP carboxylase. So PEP is reacting with carbon dioxide, oxaloacetate is formed, oxaloacetate gets converted to malate, malate diffuses into bundle sheath, it gets converted to carbon dioxide and pyruvate. Carbon dioxide takes part in the Calvin cycle, pyruvate is again going back to the mesophyll cell and converted to PEP, where this cycle continues in the mesophyll cell and Calvin cycle continues in the bundle sheath cell. So this is how the C4 plants are combating against photorespiration. Now, apart from C4 photosynthesis, there is also another photosynthetic pathway called the CAM photosynthesis or CAM plants, where the kind of photosynthesis that happens is crassulation, or the full form of CAM is crassulation acid metabolism. Now, as compared to C3 and C4, CAM plants have a totally different way and timing during which the Calvin cycle happens in them. If you see C3 and C4 plants, the stomata will be open during the night time. The morning time, and it will be less open during the night time. Whereas in CAM plants, what happens is CAM plants keep the stomata open during the night time and closed during the daytime. Now, this kind of a mechanism is to save water for these plants called CAM plants. So in order to let carbon dioxide inside, they'll keep the stomata open in the night time, during which the water loss will be less, because there is no heat as compared to the kind of heat they experience during the daytime due to the sunlight. So they keep the stomata open at night, let all the carbon dioxide enter them, and then by the daytime they'll close their stomata. And during the daytime, this Calvin cycle will happen. Now let's see, and this is how the leaf structure looks like. And if you want to relate to more, CAM plants are plants like cacti and those plants with thick fleshy leaves. So most of the plants that you find, like molasses, the plants that you find in desert regions, even pineapple is a CAM plant. So if you see, their leaves are different as compared to C3 and C4 plants. They have thick fleshy leaves, and they require their function is to store a lot of water. So in order to reduce the transpiration rate, the stomata stay open at night and closed during the day. Now, how does a photosynthesis or the Calvin cycle happen is, during the night time, like I said, stomata remain open and carbon dioxide enters. Now, when this carbon dioxide enters, it is not stored just like carbon dioxide. It is converted to oxaloacetate by the enzyme PEP carboxylase, and then oxaloacetate gets converted to malate. Malate will then be stored in the vacuole of the cell as malic acid. So when I had shown a plant cell, I had said that there are chloroplasts, there is a vacuole, there is a nuclei. So this malate, which you are familiar with now by the C4 photosynthesis, malate is not getting converted to carbon dioxide and pyruvate, but instead malate is converted to malic acid and stored in the vacuum during the night time. So during the night time, carbon dioxide is fixed. There is carbon dioxide is converted to oxaloacetate, oxaloacetate is converted to malate, and malate is stored in the vacuole as malic acid. Now, as the time passes from night to day, during the daytime, what happens is the stomata will remain open, and during this time, the malic acid which is stored in vacuoles will get converted to carbon dioxide, and this carbon dioxide is then utilized by the RUBP for completing the Calvin cycle. So during the night time, carbon dioxide fixation alone happens, and during the daytime, this fixed carbon dioxide is again converted to carbon dioxide and utilized in the Calvin cycle. So if you see the cycle over here, we see that carbon dioxide during the night time enters and everything is happening in the mesophyll cells only. There's no bundle sheath cells and all. So enters the stomata, gets converted to oxaloacetate by PEP, gets converted to malate. Malate is then converted to malic acid and stored in the nuclei. During the daytime, this malic acid is again converted back to malate, which releases carbon dioxide for the Calvin cycle to happen, and then pyruvate is again getting converted to PEP. So this is how the CAM plants have their photosynthesis occurring. So again, features of CAM: night time carbon fixation happens, daytime Calvin cycle and carbohydrate production happens. There is water. Main difference is the water conservation because of the stomatal opening at night and closure at daytime. And advantages is it is highly water use efficient and it is capable of surviving in extreme conditions. Disadvantages, the growth rate is slow because a lot of energy is required for carbon fixation. You have to take carbon dioxide, convert it to malate, convert it to malic acid, then again back to malate, and then to carbon dioxide, which is actually energy intensive. So the growth rate, the amount of carbohydrates that are synthesized is less. At the same rate, if you look at C3C4, within the same time, a lot more carbohydrates will be produced. So examples are cacti, agave, euphorbia, aloe vera, pineapples, orchids, etc. So if we compare C3, C4 and CAM, the primary environment for C3 is cool and moist. C4 plants hot and sunny, thrive in hot and sunny conditions, and arid and semi-arid regions you find CAM plants. Tomato, it is open during daytime for both C3, C4, and while CAM it is only open during night. Carbon fixation is directly happening here. C4 carbon fixation is happening through PEP, and the enzyme that is used is PEP carboxylase, and for both C4 and CAM. Then there is no separation in the photosynthetic pathway. There's light dependent and Calvin cycle, but in case of C4 and CAM, we have a spatial separation. That is, in C4 you have spatial. That is, the initial PEP reaction is happening in the mesophyll cells, and the Calvin cycle is happening in the bundle sheath cell. In CAM plants, there is no difference in the cells where it is happening, but temporal variation is there. Carbon dioxide is getting fixed in the night time, and Calvin cycle is happening in the daytime. So this will be for the module 1. I will be updating you with the module 2 lectures. This is the whole overview of photosynthesis. From next class onwards, we are going to go deeper into C3 photosynthesis alone. What are the equations required to find out the rate of C3 photosynthesis? Thank you.